Innovation and Research, Mysuru, and Department of Biotechnology, New Delhi. So for this program, we have two eminent external speakers, Dr. Jayakant Chandrasekharan, Associate Professor from SRMC Chennai. I take this opportunity to welcome him. And also we have with us Dr. Rama Kodanda, Assistant Professor from Kumara Guru College of Technology, Coimbatore. I also take this opportunity to welcome our beloved principal, Dr. S.B. Dhanbal, sir. Welcome to you, sir. I also welcome all the delegates who joined and registered for this program. So with these opening remarks, I would like to request our beloved principal, Dr. S.B. Dhanbal, sir, to kindly give his opening remarks on this program.
Thank you, Dr. Praveen. So very warm and pleasant good morning to all our honorable speakers and the distinguished participants. I bring greetings from JSS College of Pharmacy, Ooty, Rocklands, Ooty. And we are happy to host the DBT-sponsored webinar, popular lecture series on the biotechnology on the topic applications of the proteomics in biomedical drug discovery on today. And this is the first lecture being conducted from the Department of Pharmacology. So my compliments goes to Dr. Divakar for fetching the grant from the DBT. And also, I would like to express my thanks to Dr. Praveen and his team members and the research scholars and the PG students for taking lead in conducting this popular lecture series. I think good number of the participants have registered. I think it's more than 200 participants have registered and they enrolled for this program. So this shows the importance of this popular lecture series. We have with us Dr. Jayakant Chandrasekharan, the Associate Professor from SRMC Porur, and Dr. Ram Kothandan, Assistant Professor from the Kumara College of Technology, Coimbatore, as our eminent speakers to conduct today's lecture. And on behalf of the staff and students and also JSS College of Pharmacy, Ooty, and also our affiliate university, JSS Academy of Higher Research, I actually warm welcome to our honorable speakers and the distinguished participants for this program. So wish you all a good success of this program. Thank you.
Thank you, sir. Now may I request Dr. Divakar, the coordinator of this program, to take the proceeding forward.
Good morning to one and all. We have three distinguished speakers for this webinar, each one having loads of experience to share with us. Among them, the first speaker, I will give introduction to Dr. Jayakant Chandrasekharan, sir. He is an associate professor in the Department of Pharmacology, the Ramachandra Faculty of Pharmacy, Chennai. He is also a recipient of CSIR Research Fellowship and is the first rank holder in All India Entrance Exam of doctoral programs held by Pondicherry University. He had 17 publications and 1 patent to his name. His cumulative impact factor was more than 45. He is well known for his works in omics and platforms. He is one of the pharmacy personnel who is very good in applying proteomics in biomedical drug discovery. And with this brief summary, I invite him to present his talk on this today's seminar.
Dr. Jayant, you can share your screen.
Yeah, share.
Please.
Just unmute it.
Yeah.
Please also unmute.
Unmute.
Sir, we are not able to hear you. Kindly unmute, sir.
Can't hear.
Yeah, it's doing it.
I have a little bit connectivity, so rectifying it. Am I audible, sir?
Yeah.
You are audible, sir, now. Yeah.
Am I audible?
Yes, yes.
Yes.
The voice is lagging or is fine, sir?
It's fine, sir.
It's fine, it's fine.
Everything, please go ahead.
Okay. Yes, fine.
இந்த செட்டிங்ல டினர் ஓபன் பண்ணிடலாமா அந்த அட்மிட் இதோ
அது எப்படி பண்றது?
அது ஜெயக்குமார் சார் கேளும் இல்லைன்னா டிஸ்டர்ப் ஆகிட்டே இருக்கும் இல்லையா?
Is my screen visible, sir?
சரி நம்ம
Slides are not
Dr.
visible.
Divakar, is my
No, sir, it is not visible.
அந்த அட்மிட் கிரெடென்ஷியல்ஸ் எடுத்துக்கலாமா?
கிரியேட் பண்ணும்போது
இப்போ அதை எடிட் பண்ணிட்டு எடுக்க முடியாதா? நெக்ஸ்ட் ஸ்பீக்கருக்கு எடிட் பண்ணலாம் இல்லையா அந்த credentials?
மீட்டிங்ல வெளியே வரணும்.
Someone can give permission for Diva, I can give permission to share my screen. It's not
You have to give permission about it.
Yeah.
Now it's visible, sir. Now you are sharing, I think.
No, sir.
Please try again, sir.
now it's
Yeah, please, credentials. Thank you.
I'll share now.
ಇಂಟರ್ನೆಟ್ ಯಾಗ ಸ್ಲೋ ಆಗ್ತಪ್ಪ
இது இವಾಗ எல்லா presentation பண்ண முடியும். இவர் செம் எல்லாம் க்ளோஸ் பண்ணிட்டு வேணும்னா அந்த credentials சேஞ்ச் பண்ணணும்.
So please check your internet connectivity. I think my side
Try. Dr. Chandrasekhar?
Yeah, I'll try, sir, but for me, sharing it's not any.
I think your internet is slow, sir. Your voice is breaking.
I can't share the screen. Somehow
Yeah, just log out and log in one more
my
time.
voice is. Tell me 2 minutes. Yes, I'll 2 minutes.
ஓகே இல்லைன்னா உன்னுடைய ப்ரெசன்டேஷன்
Diwakar, now is my voice is audible? Is it audible? You can unmute and say. Am I audible, sir?
Yes, sir. Yes, sir.
Yes, fine, thank you. So, is my screen is visible, sir?
Yes, yes.
Yes. Sorry for the delay and interruption in the internet. Yeah. Good morning, everyone, and thank you for your warm welcome. And special
Good
thanks
morning,
to,
sir. Ashwin.
yes, special thanks to Dr. Divakar as well. So today I'm going to speak a little bit of my experience in this multi-omics dimensionality reduction in drug discovery. So if you look at the topic, it has three components. One is multi-omics, and what is the importance of dimensionality reduction, and what is exactly mean by drug discovery. So what I thought is, I thought of giving you a small introduction about how drug discovery process happens in industry, followed by work done by us on proteomics, and then the current work which I am doing on the dimensionality reduction. So there are well thought out and sought out strategies which is applied for drug discovery process. So before doing any drug discovery process, we need to understand what are the strategies followed in drug industry. So industry pipeline is very important because they have well thought out and sought out strategies. So the most important question in any kind of research or creative thinking is how they do and why they do. So the first question is, how does a chemical become a medicine? Do the process is simple? Yes. Let us look back a little bit of the history. I'll go through it very fast. So how does a chemical become a medicine? Do the process is simple? No, it's the process is not simple. You all know, you have studied in theory. It's a huge attrition process. They will encounter with lot number of failures, and and the investigators have to work according to the ICH guidelines, which are codified. We are knowingly or unknowingly embedded in this ICH for the efficacy and safety data. And need to work along with multiple departments. So the integrative approach is very important for the eventual success. So next question is the design and serendipity, how actually the drugs are designed. So from where they are getting the knowledge on the drugs, how a chemical can become a drug is a question. So from indigenous knowledge, like from treaties, we can get an idea about discovering a new drug, or from human experience, or we say serendipity or discovery by chance. And in the era of technological and biological breakthrough, we can able to integrate and able to understand the ingrained roots of the pathogenesis of a disease, which helps us to identify the key driving molecules or the targets which are responsible for the diseases. Yes, we do agree. We are in the omics era. We have humongous data. We don't know how to correlate and how to make it as a meaningful one. So this technological and biological breakthrough helps us to identify the target, and then we can specifically target those drugs which can target that, so that we can minimize the side effects and we can also improve the efficacy. So discovery to market, the economics, a small look back. You can see that from discovery to IND, it will take 5 years. From IND to NDA, it will take 6 years. And then the regulatory approval will take 1.1 years. When we put all these years cumulatively, it takes 12 years, and the cost they spend is 1.3 billion. And the success rate is very, very less because 1 in 1,000 compounds will hit the market. So this slide is very important who are doing as a PG, PhD or anyone. So where you want to work. So we need to define our ground zero. So what is the meaning? The first slide says that low validation state to high validation state. Because I see some of the students straight away go and do the in vivo experiments. Yes, we conceptualize the hypothesis. First, we have to prove our hypothesis in cell-based assay or the receptor binding test. So once your hypothesis is proven in vitro status, then we try to validate in the higher level, that is in vivo in animal model. In terms of animal model, selecting a valid animal model is important. So I have in the coming slide, you can see one classical case study of a company. And then once in animal model, the drug shows good safety and efficacy, then we translate to clinical trial from phase 1, phase 2, phase 3, and finally the drug will be launched. So the hypothesis goes from low validation state to higher validation state so that we try to prove our hypothesis and make the statement stronger and stronger. And most important thing is most of the companies work on previously validated clinical targets. So what is the meaning of this previously clinically validated targets? So 70%, which means that, for example, if we do proteomics or phosphoproteomics or genomic analysis, we try to do by taking multiple samples, and then we try to figure out what are the key drivers, and they try to validate all those things in clinical trial, and they'll claim it as yes, it has a strong association with this disease. So these clinically validated targets, most of the 70% of all marketed drugs are on already clinically validated target. So on compression, um, if anyone want to do a very good piece of work, they can work on target identification is a primary thing. They want to go for a first-in-class target. It takes 1 to 3 years. So once we identify the target, we try to do lead generation, that is by using different methods of virtual screening and cell-based assay to prove the efficacy. That's identifying EC50 or IC50 depends on agonist or antagonist. Once it is defined, we try to optimize the lead. The optimization is all about pharmacokinetic parameters. So how you can optimize the absorption, metabolism, elimination, all these things. So next phase, it goes to preclinical development, and then finally, before clinical phase, it went into the formal development, that is 4 to 8 years. So a person who plan for their PhDs, they can work exclusively on identifying the targets by applying all the technological advancements that is using the omic platform. And once the target is identified and it's a clinically validated target, then you can go for lead generation, then lead optimization, then preclinical development and formal development. So, and now the duration is almost compressed because of all this advancement. So lead generation we can able to achieve within 7 months. Lead optimization can be done in 9 months. Preclinical development can be done in a year, and formal development happens within 4 years. So within less than 7 years, we can able to hit a product. So what is the compound selection criteria? Because in pharmacological sense and in terms of chemistry sense, they have a different opinion. So uh, biologists and chemistry personnel have to work together if they really want to succeed, or you should have a very good biological or chemistry background to have to select a compound. For example, as a biologist or a pharmacologist, my aim is the target is a biological pathway that can be intercepted in some way to give useful therapeutic outcome. So we want our drug to go and bind with the target, can act as an agonist or antagonist. We always look into the intrinsic activity. Whereas in chemist sense, he always try to optimize the ADME properties. In his sense, the biological pathway can be intercepted in a useful sense by a orally active small organic molecule. So they prefer any drug which can be given orally. And interplay of this discipline leads to the eventual success. And this slide is a very, very important slide. And this is how all the industrial paradigm or in the terms of drug discovery work. And it's the pressing need to understand how we need to approach. So how to typically compound selection criteria goes on. First, we have to focus whether we are going for first in class or best in class target. So what is the meaning of first in class? You are developing a molecule for the first time for the target is called as first in class. And in terms of best in class, already for the target, there are many drugs are available in the market. So whether if you want to focus on best in class, then we need to thoroughly go through the limitations of the existing drugs which are in the market. And most important second criteria we should keep in mind that whatever compound you are going to generate is should be structurally unique. Then only we can hold IPR and intellectual property rights, and it can be patentable one also. So the first primary aim is to focus on first in class or best in class, and second thing is structurally unique molecule. The third comes the solid pharmacology. If you are working on best in class, the potency that meets or exceeds the gold standard, which is already available in the market. It should be equivalent or more than that in terms of efficacy or in terms of any of the uh, pharmacokinetic parameters. And if you look at here, the target selectivity. For example, if I work on a kinases, we know the kinase family is big. We have 113 kinase, 119 kinase family members. So since they are the family genesis, they always have the off target effects. So when you plan for a target, you need to make sure that it's more selective to the target what we have defined rather than the closely related targets, and the selectivity should be greater than 1,000 folds. And the third important thing is efficacy in relevant animal model is very, very important. And fourth, excellent drug metabolism and pharmacokinetic parameters. So in that, what are the things we need to take care of? No drug-drug interaction liabilities, and suitable for per oral dosing, and it should have limited metabolism, and the drug should have a robust efficacy in rodent autoimmune disease models. So these are the primary things which we need to keep in mind. The most important is target selectivity. It should be 1,000-fold selective when regards to the closely related targets. Now let's see about the right hand side. I have given the research testing cascade. What are the key parameters the industry will focus on? So, for example, I have found a drug. And I want to do a receptor binding assay, and it has to produce, or EC50 should be, or IC50 should be less than 10 nanomolar. Then only when we escalate the dose for the animal and the safety study, it can be convertible into drug. So our aim is um, the heat or the lead molecule should possess its EC50 or IC50 less than 10 nanomolar. And the second important thing is the solubility. The solubility need to be zero point 0.1 or zero point 0.2 milligram per ml in the pH of 7. And third important thing is you have to check the T-half in human microsomal liver enzyme, mouse and rat, and the T-half stability should be greater than 16 minutes. And also the hERG binding when compared to acimazole, the standard one, it has to bind with greater than 10 micromolar. It should not have binding ability, or if it is binding, it should bind with a greater amount of the dose. And human 5 major P450 enzyme, we have to make sure our drug is not the substrate or the inducer and inhibitor of any of the uh, microsomal 450 enzyme. And finally, the mini Ames test, it is used to identify whether the drug is mutagenic or carcinogenic in activity. So when we plan for a PhD work, or if you really want to translate whatever you have found out, the best ideal things are these are the key parameters which you should keep in mind. Then only whatever you have found out can be translated to the next level. That is the second tier studies. That's oral efficacy and dose response, and it has to possess the oral efficacy or dose response of 1 milligram per kilogram body weight, and mouse PKPD we are supposed to do, and single dose IV per oral, cardiotoxicity, CREP panel, which means the kinase panel, and the Ames mutagenicity test. So like that, we try to curate or scrutinize the drugs so that the success rate can be well and good. So this slides is a very important slide. Who are in the research of drug discovery has to keep it in mind. And what are the different challenges and opportunities in the lead generation strategy? Here I have given how actually the pipeline works. Since we have a very good virtual platform, you can use any one of the methods. You can use shapes and fragments. You can also go for scaffold morphing, virtual screening, de novo design, BIOS library, or you can take known inhibitor and you can go for pharmacophore modeling. There are different types: ligand based, receptor based, atom based, all these things, and then compound screening from the FDA approved library. So from this, you screen and you can predict, okay, with this target, it's binding good and the interactions are maintained well and good. So those compounds can be tested in biochemical or cell-based assay based on your priority. Once they are showing at the nanomolar concentration, then these complex need to be co-crystal and their interaction need to be studied. We can use NMR, you can use X-ray diffraction. Now the cryo-EM has come up so that we can stabilize the complex in 4 hours and can able to identify the key interacting patterns within 4 hours. And this cycle goes on, still we optimize the lead, and finally in vivo, in vitro, developmental criteria, whatever I have shown the tire, no, so that can be applicable, and finally we will develop a lead. So the last slide and this slides are very important to understand how the industrial drug discovery paradigm goes on. So I just want to give one of the known example how the vidagliptin is came into the market. So I'll just give you a case study kind of thing. We know that vidagliptin and allogliptin are anti-hypertensive, anti-hyperglycemic agents, anti-diabetic agents. This allogliptin. So let's see from where they got this idea and how they identify this target. So we always think that we used to get the research question from the bedside, which means we have to closely associate it with the physician. So what they have observed is when they give the oral glucose, the insulin secretion is well and good. If they give the same glucose in IV, the insulin secretion is not that much. So that's the key question they have in mind, and they put forth to the scientists. They try to do what is the difference, why it is existing. When oral glucose is given, it releases GLP-1, which triggers the insulin secretion, whereas in IV, it's not triggering the GLP-1. So now what they thought about is whether we can get an analog of GLP-1 and give to the patient. So there are few drugs on GLP-1 analog. And second thing is what they thought about is how this GLP-1 is getting metabolized, because its half-life is 1 to 2 minutes. So the DPP4 is an enzyme and it acts on this GLP-1 and metabolizing it. So what they thought about is why can't we hit and this DPP4 inhibit this activity so that the half-life of GLP-1 can be increased, and then the activity, anti-diabetic activity can be produced. So am I audible? Am I audible?
Yes, sir.
Yes, fine. Just take, yeah, fine. So what they have planned is whenever you plan an experiment, we should always keep some absolute criteria and then relative criteria. So what is the meaning of absolute criteria? So I want a drug, I want a DPP4 inhibitor. It need to be highly selective to DPP4 and it need to be very potent. And second criteria we should keep is there should not be any cytochrome P450 interaction, and it need to be suitable for once daily dosing. And fourth is the drug needs to be orally active. If I am going for best in class, it has to be a superior efficacy and it should possess safety profile. And in terms of relative criteria, since we have the key competitors like vidagliptin and sitagliptin, so the efficacy needs to be equivalent or superior than the competitors. So how they have started their work? So what they have did is they looked at the co-crystal structure and looked at the interaction patterns, because when you plan for when you look at, because people are now fascinated after COVID about docking and dynamics, but they will end up with docking score, all these things. But the most important key thing they are missing is you have to have a very good knowledge about the active site, key interacting residues, where the interaction need to be maintained. So the co-crystal, they visualize what are the co-crystal, the vidagliptin and alogliptin with DPP4, and identified key interacting residues, and the structures are depicted here, and the structure what they have found out of virtual screening is structurally unique. So yes, it can be patentable. And next complexity comes, since the DPP4 family is a dash family, they are supposed to look at the structural analogs. So these are these closed structural analogs. So as I said, the selectivity should be greater than 1,000 folds when compared to that of the structural analogs. So they are looking at what are those structural analogs and what are their biological role in relation. So FAP alpha, DPP2, DPP8, DPP9 are few of the structural analogs, and they summarize what is the biological role. So T cell apoptosis, attenuation of T cell activation. So any hindrance should never make any physiological parameters inside the body. So now, fine. So they found out lead. It's called as an allogliptin. How they proceeded with the experiment. So first and foremost aim is they went for an enzyme binding assay with their competitors as well, and they found out the drug is inhibiting at zero point 0.9 nanomolar when compared to the sitagliptin and vidagliptin. So you can see the nanomolar concentration here. So that first and foremost, they they have proven the hypothesis. Their potency when related to the existing drugs in the market is well and good. And second thing is, as I said, we should always check the selectivity with close analogous proteins, DPP2, DPP8, DPP9. The selectivity is greater than 100 fold, I mean 1 lakh folds. You can see here all. When compared to vidagliptin and sitagliptin, you can see DPP8, they have 1400 selectivity, and DPP9 81.5. So this gives why the toxicity appears with these drugs and how we can overcome all these things. So whenever you choose a drug, we should always check the selectivity with the closely related family members. And now comes as a pharmacology, how we have to do all the things. The first and foremost, they have chosen non-obese diabetic STZ, streptozotocin rat model. So we always have a debate in M-Form classroom, you know. So this alloxon is type 1 and streptozotocin is type 2. Some will deny it. But in terms of pathological or clinical perspective, we keep this model for non-obese diabetic individual. So first and foremost, they took a non-obese diabetic ST is a DAT, and they try to find out the activity. So they have given different dose of alagliptin, and DPP4 activity is measured. Yes, it's decreasing. And likewise, if DPP4 is inhibited, then second endpoint is GLP-1 has to get increased. So these are the two key endpoints they should keep in mind. So whenever you design an experiment, what is needed, that is enough to do on that. So their hypothesis is the drug will inhibit DPP4 and enhance the activity of GLP-1, that is proven in the in this model. And second thing, yes, GLP-1 is high. Now the second level of endpoint is in terms of response, it should lower the plasma glucose, and the insulin secretion should also be very high. So they went with OGTT, and they found out this drug is reducing glucose and increasing the plasma insulin. And we because we need to claim whether it is an anti-hyperglycemic agent or hypoglycemic agent. We have minor distinction between these two. Hypoglycemic agents are not adaptogenic drugs. Even if it is taken by a normogenic individual, it will reduce the blood glucose. Those are called as hypoglycemic agents. So for example, metformin, it won't reduce the blood sugar for the patient who are normal or eugenic. They don't have, their normal blood sugar will remain same even they took 500 milligram. So the next aim is whether they affect the fasting blood glucose level. So they tested in the normal SD rat and they found out it's not reducing to a greater extent. So we can claim this drug as an anti-hyperglycemic agent. So, and coming to the next, what are the animal models we should select? We have seen they have chosen STZ model and it replicates non-obese diabetic model or diabetic individual. Now they went with the DB mice. So I have given in red color. So they have a gene mutation in leptin receptor and confers the susceptibility to obesity, insulin resistant, and it's an ideal model for obesity associated with type 2 diabetes. So they went with the DB mouse and experimented all the things so that we can make a claim that first claim is it's an adaptogenic anti-hyperglycemic agent. It can be used for non-obese diabetic patients. Next, they have used DB mice and confirmed their hypothesis. It can be given for obese diabetic individual as well. And similarly, they have tested all the hypotheses, DPP4 level, GLP-1 level, all the things. And further, they want to check on the pre-diabetic model also. This OB mice model is a very good model for pre-diabetic model. I hope we have three categories. One is no normogenic, normal. There is no reduction in blood glucose. They are normal individual. Next is they are pre-diabetics. They have the fasting blood glucose level of 180, and then we term it as third one is the diabetic individual. They have more than 180. So whether this drug can be given for pre-diabetic patients or not, for that they chose the OB mouse model, and in that also they check. And you can see here these OB mice also lack the leptin gene. These mice display progressive food intake, resistant, but there is a moderately increase in blood glucose level where we can correlate with the clinical pathogenesis of pre-diabetic one. And this is how they have designed the experiment. And since it's an insulin, they also counter check whether the axis is getting altered or not. So they also check the plasma glucagon level. And those are the pharmacological endpoints what they have checked. They checked in non-obese STZ mouse model, and second thing is they checked in DB, and the third is OB. So it can be used for non-obese diabetic, obese diabetic individual, and third is it can also be used for pre-diabetic patients. And finally, when we look into the pathological endpoint, which we used to say in terms whether it increases or restores um, the intensity of insulin in the beta cells or not. So pancreatic insulin content restored with the drug or not is done at the level of the pathological endpoint, and they found out yes, pathologically it is also working. So why I started with the drug discovery platform is to nurture you how actually logically or rationally we should go and approach what is the meaning of valid animal model with a classical example. Now let us speak about the work which is done by us on proteomics. So what are the things which we have identified, and this paper have been published in Biofactor 6.438. And let me explain to you what we have did actually. So this paper is we worked on in vitro model for acute lung injury. It's painful conditions or the terminally end conditions, acute lung injury. So in cell line, we mimic the same model by supplementing TGF beta 1, and the known. We also checked how the curcumin is doing its work. So we have a control, and then we have a cell line treated with the cell line we used is A549 cells and TGF beta 1 plus curcumin. So once the treatment is done, we isolated the protein, went for tryptin peptide digestion, and then fractionation, and then mass spectroscopy based proteomic approach. So here we did label-free proteomics, and then you can see here how many proteins are identified, and what is the number of proteins which are upregulated in TGF beta 1 treatment group, and what are the proteins which are down regulated, what are the proteins which are upregulated with when we by giving curcumin as an interventional one, and what are the proteins which are down regulated. So this gives the list. So once you perform a proteomics by isolating the proteins and then doing in a very good mass spec and either label or label free method, you will get the n number of proteins. Next complexity comes how to look at the data, because this data is humongous. We have nearly 3,000 proteins. How to make this data as a meaningful one? You can see here what we did. In left hand side, we have shown something called as heat map analysis, which explains the intensity of the expression. You should keep in mind that the proteomics is a semi-quantitative analysis. Whatever you are finding has to get validated in terms of absolute quantification using Western blot or ELISA. So so we have taken differentially regulated proteins, and then in control group, what are the proteins in TGF beta 1 and TGF beta 1 plus curcumin treatment groups. The proteins which are highlighted in red are differentially regulated. So in that we found something A COX1, and so we thought of validating it in through fluorescence staining, and we also found that the treatment with curcumin is enhancing it. So next our aim is, here I have shown the nearly 11 to 12 differentially regulated proteins with and there we also check their fold change in the mRNA level, and few have been taken forward for Western blot analysis, and few are checked whether it's a druggable target or not, and we also docked curcumin with that, and we found out whether it is really binding with this or all the things. So if I want to give a PhD for the student, I can give each target to one student, and they can work comprehensively on identifying or developing a drug for that. So this is a little bit a brief information about proteomics. And finally, let me come to the dimensionality reduction. So this is presently I'm working on, where I have implemented machine learning and AI model to it. So we plan to identify a selective CAM-KK2 inhibitor. So we know that CAM-KK2 is an emerging target, and there are 8 chemotypes were patented. When we did a patent review, we found that there are 8 chemotypes, and there is a known experimental inhibitors, that is STO609, and there is no investigational drugs till now, and nothing is in market as well. So these are few of the key things which we found out. And when we look into the complexity of this is why there is a complexity exists in going for a selective CAMKK2 inhibitor is, since CAMKK2 belong to the kinase family, selectivity obviously matters. So it has to be more selective to CAMKK2. So now we thought about, yes, we have Schrodinger, drug discovery, all these platforms, and how we can use all this platform, what are the limitations of these platforms, all these things we analyze. And also we analyze the pitfalls of existing visual screening approach. We have natural compound library, we have commercial compound libraries. In terms of natural compound library, source isolation, identification, chemical modifications are bit challenging. And in terms of commercial compound libraries, you can end up with the same core scaffold, lack of molecular diversity. So the first thing is what we keep in our mind is there a way to produce a molecular diversified compounds, and it should also have a synthetic feasibility. We need to achieve the synthesis in few steps without any difficulty is a question which is hindering our mind. So what we looked at is we looked into chem informatics platform, which are open source, that is RDKit, which is more famous in drug discovery portfolio. And also we use StarDrug platform for rule-based transformation to produce chemically diversified compounds by using NOVA, Bioster, ring replacement, conformational constraint, and then reversible dimerization. So it integrates all these things and it will give up a structurally unique molecule from the molecule we provide to the system. So, so when we thought about how to overcome all these limitations, we thought of using RDKit and then start our platform for that. So in RDKit, what are the things we can able to perform? So we always keep an when we using star draft, when we found out molecular diversified compound, we need to filter it. So we kept log p, a coefficient, an acceptable one. I am not showing the value here, and hydrogen bond donors, total polar surface area, qualitative drug likeliness, MCE 18 for structural diversity. The values are very important, and also we kept Lipinski rule of 5, and the final one is synthetic accessibility score. It ranges from 1 to 10. If we get a synthetic accessibility score of 1 to 4, then it's very easy to synthesize. So all these pre-filters we kept in our DKIT program. And second important thing what we did is, uh, so we speak about ligand based or we develop pharmacophore, but in recent trends, they are using extended connectivity fingerprints or Morgan fingerprint, we used to say. So what we try to do is we will try to, for example, if a structure I found out means I try to dissect that structure into vector maps into 512 bits. So like this, it will quantify with each value, with each bits of the vector. So that this is called as topological fingerprinting. We utilize that. So once topological printing is done, next we subjected all these 512 fingerprint vector maps for T distributed sociostatic neighboring embedding, that is TSNE, is a dimensionality reduction visualization tool where we can it will cluster because it has to reduce 512 dimension into cluster and it will give a correct picture. After that, we go for docking dynamics, all this portfolio. So here is the workflow for you. If anybody is interested, can follow this workflow. Um, so what we did is we thought of taking a starting material as an FDA approved drug because it's available. We can for synthesis if we are going, we can use this as a starting material. So after getting FDA approved drugs, we did an SP docking with CAMKK to active site, and here I have given how we chose the top 10 or the top 10 docking drugs is based on the docking score. And second is key interacting residue. Most of the people leave this. We have to see what are the key interacting residue. Ligand efficacy score will also matters a lot. So once I found out top 10, I have to check with the kinome database, which means there are few databases. If you are interested, I can give you. So where we have to go and put all those FDA approved drugs which we got, and we try to find out is any of the drugs have been shown to possess cross reactivity with CAMKK2. So we found few. So it was interesting. And then from the top 10 doctor drugs, we transferred those 10 into set 1 analogs. Our first level of transformation was done using applying these two filtering criterias. First, rule-based enumeration was done on the 10 doctor drugs, and we made it as an analog. Nearly we got around 50,000, and by applying RD kit filtering criteria, as I mentioned in the last slide, we applied that and we came up with the top 10 doctor compounds from the transformation 1 analog. So this top 10 is further, it went for the second level of transformation, and this second level of transformation from the first level of transformation and also from the top 10 FDA drugs, we did ECFP4, that is fingerprint analysis of this all nearly 55,000 compounds. We converted them into 512 vector maps, and then we also did a TSNE analysis, and from that we chose yeah, excellent complex for molecular dynamics. So here I have given few of the details on the in slide. I didn't give any of the structures here. So top 10 FDA approved drugs based on kinome database, whether they have any cross reactivity with CAMKK2. And then we did a first level of transformation using a short drop. And here you can see the ligand score, and then the docking score is not mentioned here. I think it's not highlighted. And also in set 2 analog, from the set 1 top 10 we transformed again to set 2 analog. We did that same kind of docking and pre-filtration, RD kit analysis, all these things, and we found out a compound which is transferred from the drug ruca rarib, from set 1 to set 2. Here you can see minus 11.034 is having a very good docking score. The interactions are maintained, and then ligand efficacy is also good. If you look at log p value is also 2.91. The molecular weight is also in the very good way, and the synthesis susceptibility score is only 3. So this is how the dimensionality reduction we try to do it. So we will give the vector map of top uh, FDA approved drugs, first round of analogs, and then second round of analogs. And here I have mentioned, and most of this red one represent the FDA approved drug, the top one, that is Ruca Parib, and the green represent the first round analogs, and sorry, the blue represent the first round analogs, and green represent the top analogs, and most of the top analogs are lying here. So if you do any of the structural modification with rucaparib, or if we synthesize this compound, this will possess a very good activity, was the prediction which was made. And this gives, parallelly we develop a structure. We, after transforming to the second analog, you can see the structure is not available in chembel, it is not available in binding db, pdp also it is not available, drug bank. So it's very good, well and good, that we obtained a structurally diversified compound. And here also we have shown where the modification is needed from the parent drug to make it as a structurally um, diversified compound. And these are the docking scores, and you can see we performed 250 nanoseconds of dynamics, and the dynamics was excellent because the root mean square RMSD of ligand is within 1.5, and RMSD of the protein is within the range of 3, which is an acceptable criteria for going forward with a very good prediction value. So now we have synthesized that compound, and it's showing the activity of CAMKK2 inhibitor in nanomolar. So these are in the process. So this is all about what I want to explain for you today. So I guess I covered a little bit of my experience and expertise in this. If you have, if you are interested in learning RDKit or machine learning, how we did this process, you can very well contact me at this email ID, and my phone number is also given here. I'm ready to help you, and thank you for listening. So I'll end with this quote: understanding the basic is easy, appreciating the nuances takes years. And thank you so much for listening to me.
Is there? Thank you, Chandrasekharan sir, Chekan sir,
Thank you. Thank you.
for a wonderful presentation and a very good introduction on how to apply dimension reduction in the drug discovery. Audience, if you have any questions, please type it in the text box available, please. Audience, if you have any queries, you please type it in the text box so that I can, Chandrasekharan sir can answer your questions. What?
There are a few raising the hands, I guess.
Yeah, Madeline has raised.
No, you need to type it in the text box. See, there is no need to raise your hands. If you have any doubts, please type your text or unmute your mic and then you can speak. Okay, sir. Then there is no question, sir.
Yeah. You can post your questions very well.
Okay.
And I can help you in that, how to run RDK, Anaconda, Jupyter program for machine learning kind of thing.
Okay, sir.
So thank you for the opportunity provided. I must thank um, Dr. Divakar for the opportunity rendered. And I also thank HOD of Pharmacology Department, Praveen sir. Thank you so much for the invite. And I also thank the principal of JSS Ooty for providing me the opportunity to showcase my expertise at that level. Thank you so much.
Jayakant sir, thank you so much. And we are really happy that you have given a very wonderful talk. Thank you so much.
Thank you.
Hopefully, we'll be able to call you in person sometime.
Yes, definitely.
Please do accept our invitation in the future.
Thank you.
Thank you,
so
sir.
much.
Thank you.
Thank you.
Divakar, you can go ahead.
Okay, sir. So our next speaker is Dr. Ram Kodandan. Okay, he will join us in few minutes. See, you all you see. I think Sir has joined now. Good morning, Ram sir.
Yeah. Can you hear me?
Yes, sir. We can hear you, sir.
Yeah, just give me a minute. I'll just finish.
Sure, sir. Sure, sir. Take your time, sir.
Yeah, sure.
Sir, good morning, sir. Shall we start, sir? Ram, sir?
Yes, we can start.
Our next speaker for this webinar is Dr. Ram Kodandan. He is an assistant professor 3 in the Department of Biotechnology with a specialization in computational biology. He completed his PhD from BITS Pilani, Rajasthan, and earned his master degree in master in biotechnology from Kumaraguru College of Technology, Coimbatore. He is a CSIR Research Fellow and has been awarded the Young Scientist Award by TNSCST 2022. His research interest includes understanding the molecular mechanism of infectious diseases and has a year of experience in molecular docking and molecular dynamics. He has a keen interest in machine learning algorithms and has developed a tool for detection of oncomics and has solved various biological problems with artificial intelligence-based solution. He holds 30 publications to his credit. He is a prominent reviewer in many indexed national and international journals. And he is a very eminent personality. He has loads of experience in artificial intelligence, and he is here with us to share his experience. Now I invite Dr. Ram Kodandan sir to start his presentation.
Okay, thank you, Divakar. That was a great introduction, actually. Good morning, everyone. I'm Dr. Ram. I'm from Kumaraguru College of Technology, Coimbatore here. Just a minute, I'll present my slide over here. Divakar, or can you present my slide and send you because I have a problem here.
Okay, sir. You can share in the email, sir. We'll present it.
Yeah, just.
Sir, you please share your presentation in Ooty pharmacology email, sir.
Yeah, just a minute. Because I'm using a different browser, so I was not able to. Just a minute. I'll go out and come back. Okay.
Sure,
Because
sir,
yeah.
Okay, sir. Okay. No, he knows. I have invited. Dear participants, please wait for a couple of minutes. Our speaker will be joining anytime now. Sir, this is Divakar, sir. Your screen is visible to us, sir.
Divakar, can you hear me?
Yeah, sir. We can hear you, and then we can also see your screen, sir. You can go ahead.
Yeah, so this session, at the end of the session, you'll be able to understand what is sequence homology, how it is being used in structure prediction and everything. I'll be showing a short demo on how to predict from structure to from sequence from structure. To start, the entire slide is divided into 3 sessions here. I'll start on with a small basics of here, and then I'll proceed with a demo of how a sequence to structure is being predicted, and the last 5 minutes is for Q&A sessions. To start with, I have a question. You can answer this in your chat session, starting with a why. Which one is faster and why it is designed so? I want to just know what's the answer for this. You can put up in the chat actually. Which one is faster actually? Whether the F1 car is faster or the road car is faster, and why it is designed so? Anyone can answer also.
F1 car, sir.
F1 cars are faster, but why it is designed so? Only one people can sit in this, and it is not so comfortable also. Is there any special purpose for that? Anyone? Just a guess. Okay, I'll go with it. Okay, F1 cars are meant for just for going faster, whereas your family cars, road cars and everything is for comfort and everything. Okay, so each one has its own purpose, and that's why it has been designed so. The same way the proteins, okay, the same way the proteins are designed in nature for the purpose and the purpose only. So if you want a protein to do some job or something like that, it will be evolved in such a manner so that the particular protein will do its job correctly. In order to evolve a particular protein, there are different processes by which a protein can evolve. The first one would be a random mutation. It's just like going for the best of it. Okay, so you can go with the mutagenesis and everything. So every time the protein gets refined and repeated mutation gets on it, so that it performs its work perfectly. So evolution takes care of everything. Whatever the processes happens within this particular protein, say a mutagenesis or something like that, the evolution takes care of it. So for an example, when we started with the COVID season, we had only one strain out of it. and we had almost found the medicine for it within the 3 months. But after a few months, we heard the story. Every time the particular COVID is being mutated every time. So what is this particular mutation? Why it is so being mutated also? The main reason would be this particular COVID started mutating of its own to avoid every drug. So any drug that has been decided developed against that particular protein was getting reducted, or it is not been working at all. So what happens is that particular organism has a very fast mutation rate, and it was evolving in such a reason. Okay, so whenever we start with a protein or something like that, we have to have a very common question whether the structure, the protein structure, is going to have any effect on the function or not. For the answer would be yes, the structure of the protein determines the function of the protein also. Okay, so that is why we are going for every time whenever we go for structure prediction, or the need for the structure prediction arises only for the identifying of function. Okay, the question here would be how do we feel it, how do we identify, how do we do it actually, how do we do the structure prediction, how do we find it, and how everything. In order to answer this particular question, we have to know a little bit upon homology. Okay, homology is look alike. Okay, so the question here would be, do you all think that we humans share a very common trait with other animals? We know that we share a very common trait with that of chimpanzees, bat, and cow. And what we share is a homology, a function. So consider as chimpanzee, we both walk almost similarly. We walk only thing walk similarly. Only thing between the chimpanzee and the human is they don't talk and we talk a lot. That is the things. Okay. So we share a common domain or a functional part with that of a chimpanzee. So this will help us to find this, compare and finding a structure of the things. Okay. So here comes the actual part of the seminar with the homology in protein. Okay, what do we mean by homology? Means any sequences. If we are going for any sequences, if when it shares a common ancestry, say it commons, for example, take hemoglobin and myoglobin. It will be sharing a common ancestor of its own, and it will have perform the same functionalities. Okay, suppose two proteins are said to be homologous, and it will be having similar kind of structures and similar kind of functions. That is what a homology is. The term homology also has some in-depth understanding, saying that what is the identity, similarity, and conservation. So before going for the homology, we have to understand what homology, or there are two kinds of homologous parameters over here. The first one would be an orthologous, and the second one would be a paralogous. Okay, and when you say a sequence is homologous and it is being shared by a common ancestry, you can define them whether it is having a, say, from the same species or from a different species. Okay, so if it from the same species, different species that arose from a common ancestor during a speciation, it will be told as orthologs. And when it is from a different species, homologous is from a different species and it arises from a gene duplication event, it is called a parallax. Both of them do the same functionality only, but the thing is they will be they arises because either because of speciation or because of a gene duplication. So when we go for a homologous thing, we have to consider whether we are going to compare its sequence homologs or we are going to compare its structural homologs. Okay, the same example, when we compare hemoglobin of human and the myoglobin of human, both are globulin families only. Okay, both are globulin families. When we compare them sequentially, they only share only 22% of the amino acids. Okay, but when you take up the structure, you know the function of the hemoglobin. It attaches heme and carries out oxygen in your blood. Okay, so it attaches heme. So when you take up the structure over here, so here in this structure, the blue color ones are the myoglobin. Okay, and the hemoglobin are the clay color ones, and the heme here it is marked in red color. So in both of them, you could clearly see that the binding parts are similar. Both the heme is bounded to the same functional part of the parts of the protein. So both of them function together. But when you come to the sequential part, it is sharing only 22%. Then how come you say, sir, they both are homologous protein or something like that? Yes, this is a very contradiction part. in bioinformatics, when you have if you don't have sequence homology also, there are structural homology, and you can easily find out functional part of it. Okay, so even if the protein fails to share a sequence homology, you can predict their structures and find whether they share a functional domain, same functional domain. Like here, the functional domain part is heme binding domain, whereas the heme binding domain is histidine over here in all the proteins here. So when they share the common protein functional domain, they can perform the same functionalities. So predicting the structure of the protein will help us to understand what is its function. With the sequence alone, we won't be able to understand what is its function. So this is what the entire section is about. So how do we do it, sir? Usually the function of the protein, the structure of the protein can be predicted by a number of experimental methods over here. The things are here you can predict them using crystallography, NMR, cryo-EM. But all these processes have a very downside. The only downside is time and money. If you need to do a crystallography, say for example, at the lab where I work for my PhD, they did for a crystallography of Vibrio cholerae, infectious disease. They took up a quorum sensing protein and they were working on it. Okay, so they were able to identify its structure only after 4 years of time. Okay, it took almost 4 years. Why this 4 years means? You might have known protein doesn't function as a single entity. It will be functioning as a group of proteins. So before going for crystallography, you have to extract the protein as a single entity, make a crystal out of it, find its structure, solve crystallographic data, and then come to a conclusion saying that extra crystallographic data. So it took almost 4 years to complete for us. Okay, for a single protein to identify its structure and everything, we took out almost 4 years. And moreover, we had n number of disadvantages also. This particular protein was very unstable over temperatures, where we were have to work within less temperature zones like -4, -6. Only we have to sit and extract the protein. So this much amount of things are there. So this will take more amount of time. But now using bioinformatics tool, within 15 minutes or 16 minutes, we will be able to find the structure. We'll be not able to get the full complete structure, but we'll have an idea how this particular structure will look at, how many alpha helix will be there, how many beta helix. So in order to do that, you just simply extract the protein, go for a mass spectroscopy, use that mass spectroscopy data as an input for your bioinformatics tools. Okay, for bioinformatics, for theoretical predictions, you can go either for modeler, okay, homology modeler. That is what we are going to discuss today. Or else you can go for a tool called Rosetta. I'll tell you what this Rosetta will do in the last slides, but we'll be looking upon this model alone right now, the homology modeling. So when a protein, so suppose if you want to do a homology modeling, the key ingredients would be, there are 3 ingredients for that. You should be having these kind of tools in your machine. Both this homology modeling can be done both in Windows machine or Linux machine. Okay. The prerequisite for running this would be, you should be having a Python 3 with IDE installed within you. And the third thing would be, you should have a program called Modeler. Now the modeler has rose up to 10.4. So, and you should have, fifth one, you should have a Notepad++ Jenny or something. This is just to edit the code. And the last one would be, you should have a molecular visualization tool, a PyMol, and possibly a usage of Chimera. Okay. For this particular modeler tool is free for academic purpose. If you register in this particular download site, and they'll be sending you this license key as modeler range, and you can just install it. This is free for academic use, and it has some limitations also, because it's free, and there's no full version for it. It runs by iteration. So if you want to get a pure model, a good model, you have to run so many iterations out of it. Okay. So there are 5 main important steps before we start your homology modeling. What are the ingredients that we should have, and what are the steps actually it is going into for? The 5 main steps would be, the first step would be, you should have a sequence of what protein you are going to have. Okay. And you should have a PDB database against which you have to search for homologs. So simply with the sequence alone, you can't build it. Okay. You should be comparing it. I think you should be aware about BLAST searches and everything. So you can use the BLAST and find homologous sequences and structures, so that you can create your own homologous structure out of this particular protein. So the first thing would be you should have a sequence for your particular protein. Usually we call this particular sequence as a query sequence. Okay. The second thing is you should have a PDB database downloaded to your machine so that you search against a PDB database out of it. The second step would be once your PDB database is ready and your structures are ready, your sequences are ready, you should be able to select the template from which you are going to copy. Okay, so you should be selecting a structure which is close resemblance to yours. For example, if you are going, suppose we keep an example like this, you are going to model the myoglobin structure. The closest structure would be hemoglobin of humans. Okay, so you'll be able to do this with the help of BLAST. So second step would be you'll be selecting it particularly. And the third step would be aligning this particular template with that of your query sequence. And fourth step would be model building. And fifth step would be evaluation. So if you are doing a model homology modeling, and after doing model evaluation, if there would be scores, that would be Ramchandran plot, process scores and everything. If it is not correct, and you have to go again for the step one. This iteration would be done until you get a exactly good one. Okay. To start with, as I told, you should be having a query sequence. I'll just show you the site where you will be downloading all those things so that it will be easy for you. You can just go to Modeler. So this is the site where you have to download. Just go for download 64. Most probably all students will be using Windows machine only. So go for downloads here and go for 64-bit here. Okay, 64-bit Windows. Okay, so this particular Windows, you can download it and it's straight away installation. It will be asking for the key alone. Just give this particular license key as your input and you start it with it. Okay, so once you download, after that you need codes for running. As I told, it runs entirely on Python only, so you need codes for running everything. So these codes can be obtained in the tutorial section in this same site over here. The first thing is if you're going for the first time as modeler, just go for basic modeling only. Just go and download this basic modeler and download this zip file. Okay, I have already downloaded and I'll just show it for you. Okay, so once you downloaded, you'll be having these many files out of it. You'll be having all Python codes and subordinate files for this. You should be having your files in FASTA format. The first thing is, where do I find FASTA file format for my file? Let's say you are going to find a FASTA file format. Just okay. Suppose if you want to find any file, FASTA file format, if you're not able to, there are people who will work with already existing entries and everything. And if you're not able to do mass spectroscopy data, it's well and good. Most of the proteins whose structures are not been predicted, but their sequences are deposited in UniProt. So you download your required files from UniProt. Okay, just click on here and you will have your FASTA sequence downloadable over here. So there are two kinds of FASTA formats usually being used. Better go for canonical FASTA. So just to show you what is FASTA file format, and just opening it up here. So the FASTA file format usually starts with a greater than symbol, and then the naming convention runs up in the first line, and then your protein sequences, whichever it's been obtained from the mass spectroscopy data, is here. Okay, so this is the file, this is the input for our modeler. So this input has to be converted to a modeler format. This modeler format is nothing but usually this particular input here. Okay, this modeler format is in the format of ALI format, so you have to convert this particular FASTA file into ALI format. Where do I find this ALI format? It's found already in the basic examples that's been given here. Just go for ALI. It will be here. Just open up in Notepad anything. So it's similar to that of your FASTA file format only, but there are certain changes. There is a second line over here. Just copy it and keep it as such over the new FASTA file format. So this ALI format starts with the code called P1, indicating that this is a protein file. Okay, then this is your naming convention. I'm just considering lactose dehydrogenase for my structure prediction over here. So it's Trichoderma viridae lactose dehydrogenase. So the naming conversion, you can give your name also, whatever it is. So whatever the name you give in this first line should be your file name. If you are giving your name as RAM here, the file name should be ram.ali here. If you are giving different names, and it won't be reading them. Okay. So the first precautionary caution would be here. So you give P1 semicolon and then naming convention for your file as well as sequence, and then copy this entire secondary line and just try changing whatever the name you are given in this TBLH. Okay, so once you change up here, and you don't need to change anything else after that. Only one secondary precautionary constant would be, you should be giving a star symbol at the end of the file, indicating that your files have been ended. End of file. Okay, star indicates end of file, so that the interpreter won't be reading anything, gaps or anything over there. Even if a single gap is being left over here, your structure would be distress. Okay, the what are the predicted structure would be at distress. So after saving, changing this particular ALI file, the second step would be, you need, for this particular first step, you need one code, build profile.py, which will be found within the basic examples. As I told, it is searched against a structural database. So pdb95.bin is your structural database. So we'll start running this code for you so that we'll see what is the result that has been coming. I'm running in terminal machines, so I use Linux. You can run the same thing in your Windows also. So just before running this, you have to change something in your build profile.py. Okay, you have to change a single line. See here, you have to change line number 23 as your input file. If your file name is ram.ali, change this line number 23 in build profile.py into ram.ali. I'm just saving here tvlh.ali, so the same file can be used here. So I'm just running my file, which is already been done. Okay. So once you run, you need Python for this. Without Python, you will be ending up in errors a lot. So you install Python 3 before running this model. Yes, our things are done. So once step 1 is done, so see here, for every code run, there will be an output called log file. Okay, just open up this log file, and you will be able to. The main aim of this first step is to identify what all structures that is homologous to my sequence. Okay, I am comparing my sequence with the structural database and finding out what are homologous structures from which I can copy the structural information to my protein of interest, whatever the query I'm giving. So open up the log file over here and then go down a little bit, and you can see the entries over here. These are the top almost 20 to 30 entries will be there. Just consider the first 5 entries alone so that it will be used for it. These entries can be selected on 2 criteria. If you're done BLAST already, you should be able to understand what is E value. Okay, if the E value is closer to 0, then it is very homologous structure. If the E value is closer to 1, it is a divergent structure. Okay, so in this column, it is based on E values only. I will be just picking up all the structures whose E value is equal to 0. See here, a protein can have multi-chains. So in this particular protein, zooming out for your sake, so in this particular 1B8P, it is also marked that only chain A is much homologous to this and not chain B. It will be on this is the advantages of modular search over here. If you are doing regular BLAST, you won't be able to see all this particular thing. So you usually prefer to download this PDB95 and run this particular things. Okay. So you'll be getting a result like this. So you have to download all these 5 structures from RCSP, where you are already, these structures are already predicted, not predicted, solved, cryptographically solved, and this will serve as a template for my next step. Okay, suppose I am going to do 1 bbm or something like that. Just search the structure and download its PDB format alone. Go for downloads here and click on PDB format. Once this is been done, so you have to download for all 5 things, and you have to start for the go for the next step. Okay. So the second step would be you have selected your PDB, and the code record for the second step would be compare.py. So now I have received 5 best structures over here. See here, I have received 5 best structures over here. I have downloaded already here. But I must be thinking about one thing: which structure is the best structure among all these 5, and which one will be suiting as a best template for my model. Okay, for this I have to compare. So in this compare.py, I'll be mentioning all the 5. Here I have taken 6 out of here. So I have taken all the 6 structures over here, and then I have started doing my things. Okay, so I'll be entering my structures input over here, and then I'll be just line number 5 and 6 I have to edit based on the outputs that is given from the first step. I'll be editing all the structure information over here, and then running this particular just I have to go to step number 2. It's running compare.py. Okay. So So go for the last step in compare.py. You will be having a distance matrix wherein it will tell which one you can choose for your templates. Okay, so this is your distance matrix which has been compared within your best 5-6 structures over here. Go for to choose the best structure out of it. Go for highly resolved structure. Here 1.8 resolution is the best as far as I consider, because in terms of resolution, the lowest resolution is the best resolved structure. If you go for highest resolved structures, you may miss up some amount of sequence over there. So the lowest resolved structure is here 1.8 here is the best structure. So I'll be considering 1 BDMA as my template for the next step. Okay, so step number 3 would be, so I have chosen 1 BDMA at resolution 1.8 as the best template for my structural prediction. So the next step, okay, next step is to align your things. Okay, for alignment, you have to have codes. For alignment, you have to have a code called align2d.py. Now I have my template structure, I have my query sequence, and I have my code for running my alignments. You are just aligning your sequence, query sequence, along with that of your template sequence 1 BDMA, so that you copy all the structural features from 1 BDMA for your predicted structure. Okay, so what we will do is we will be editing this code over here. Okay, here in line number 5, as I told, just in line number 5, just put 1 BDM and it's A chain A over here, and in line number 6 also you put up 1 BDMA, and you mention this is the atom file. I have downloaded 1 BDM.pdb already, and this is my query sequence, what TBLDH, for which I have to predict my structure. Okay, so in this particular align2d.py, I'll be generating 2 results. Okay, one result is a sequential result, and the second result is a structural comparison result. The first dot .ali is the sequential comparison result, and .pap is the structural information research. The thing here would be it's better to you can name whatever even 1, 2, 3 that ALI also you can name. It's the best to thing is after a few weeks or so if you would go and see you won't be remember which structure you are taken. So the best thing is put a query sequence and the structure template structure name dot ALI. For this will be easier for identification even if you take and see after a week or so. So we'll run this particular code. So it will generate two files as I told. It will generate pvldh.pm on pdma.ali and PFP. Now we are ready for construction of your from query sequence to three-dimensional structure of my protein. So how do I do it? For this, I need the code called model single.py. Okay, here, so see, it has been noted that modeler also allows to use the multiple template for building up your models. Okay, so here I'm using only single model as a, this is a basic example, so I'm using a single model template as a methods here. So the code that is being used is model single.py, and the input file that I required would be .ali file from the previous align2d.py and .pap structural file from the previous structure. So we'll start running this. It will take at least 4 to 5 minutes to complete based on the machine configuration. So let me wait for this. Meantime, I'll tell you what are
Your
things
voice is
has to be.
breaking, sir.
It's audible right now, or it's still breaking?
Yes, now okay, sir.
Yeah, okay. So after this, let the model comes up. It will take another 5 to 6 minutes to build a model. We don't want to waste time. You'll be getting a time output like this. Okay, once your model single.py is been done, open up your model single log file. There will be a log file over here. In this log file, come to the last part of the log file, and you'll be having a table like this. Okay, you will be building 5 different models for a single template. So just by chance, we are building 5 building. Okay, for this, just look for it. Okay, just look for this table here. There are 3 scoring patterns. Okay, if you're done docking already or blast like things or something like that, there will be scoring for selecting your sequence. The same kind of thing here for modeler. You have mole PDF scores, dope scores, and GA341. For my, usually I'll go for lowest dope score values. I've written over here, lowest dope scores is the best one. Okay, so what I will do is the model 5. So it will generate 5 models over here. Okay, that's been done. It's still running. Okay. So there will be 5 models built up here. So I'm taking the lowest dope score value. See here, minus 37406 is the lowest score. So I'm taking this as my perfect model, and I will be going for model evaluation. Okay, model evaluation is nothing but I'll be checking the Ramachandran plot over here. I'll be going for Ramachandran. There are different methods by which you can evaluate your models. Okay, here even you can compare your model with that of your template that you have taken. Here the template that I'm taken was 1BDMA. I'll be checking up the alignment position scores, whether here see the red color is your model, whichever which I'm building. That TV 5th model I have plotted over here. So this model where along with that of the template is 1BDMA. Okay, so what happens is you can see there are deviations over here. Can you see very clearly in the 150th position over here, 150th position over here, you can see some clear deviations over there. So your structure, every time whenever you are copying your structure or something like that, you should be thinking of only one factor. There is a factor called stereochemical properties of amino acid. Not all amino acid will roll up so quickly as that of your template, and you can't copy exactly the stereochemical properties of a template. Okay, so there will be errors. So how do we reduce these errors? So these errors are very common over here. Mostly these errors arise because comes in the loop structures. Okay, look, if you're comparing a protein and everything, this low, see here, I have done for exactly, this is a plot over there. I have done structurally to show where the error has occurred. This blue color is the model that we have built up, okay, and the template is 1 BDMA. See the red color marks over here. I told you, no, the 150th question over there. So there you can see that this error over here. This errors arise because of many things, okay, mainly because of stereochemical properties of your amino acid. So how do we refine it? You can refine it. I'll tell you the refinement methods also. So that you can refine it easily using three methods over here. Or else the error might have occurred. You might have taken a wrong template. Okay, you might have taken a very low resolved X-ray crystallography template as your template. Okay, the second thing is you can improve your model based on multi-template model. This model, whatever we did, was a single template model. Okay, you can do it using multi-template model. Obviously you will get a best score out of it. Or else you can go for iterative modeling. It's a very time consuming process, but you, at the end of the thing, it may take a day or so to complete the entire process. So at the end of the day, you'll be able to get a better model than the single template model. Or else there is another procedure called loop refinement. Okay, what we will do is each we'll be looking out this particular loop and what is the constituents of the loop. We'll be checking, let's say, let the loop consist of alanine or glycine or something like that. We'll start mutating each and everything and fit up the best stereochemical property based amino acids. That's a very tedious process. There are things that can be done online also. There are tools like mod refine to do this particular tool automatically on the online sites. Okay, so using this refinements method, you can build up a best model out of it. Let me see whether yes, our models are done. Let me go for step 4. Yeah, see here there are 5 models that have been built, out of which I already have done. I have seen the scores also. The scores would be here in this model single.log. Just go for the last. Here you will be having this particular table. As I told, this table is the Bible for us to select for the template. Okay, as I told, this minus 37406, 37,004 dope scores. You can even, hello, even you, yeah, any disturbance, anything? Okay, I think some disturbance. Okay. The things are, you can see this particular dope score. You can even consider this GA341 was 1. You can't go for this GA41 obviously. Mold PDF score is a empirical score. It's a theoretical based score. Don't go for it. Usually go for energy based score. Dope score is an energy based score. Better go for dope scores in modeler for selecting your final model. So once your model has been done, go for a server called saves. Okay, go for a server called saves, and here you can verify your model using 5 different tools over here. There is error, verify 3D proof, and even project. Project is what your Ramachandran plot, where you plot your Ramachandran plot. To be very simple, I have done Ramachandran plot for you. We'll see how much amount of allowed region and disallowed regions are being there. So this is the Ramachandran plot of the structure, whatever been taken as the best model out of. We have taken structure number 5 as the best model, and we have plotted this Ramachandran plot for this. You can see alanine 98 is out of scope from everything else. It is the only amino acid out of it. Okay, and this is the plot statistics that has been done. According to plot statistics, there are no disallowed regions at all, because usually alanine is been not considered at all in this particular thing. So if you are getting, you should be getting 2% and less in your disallowed region. If you're getting more than 2%, then you have to go for multi-template model and iterative processing and everything. If your model is only consider this disallowed region alone, less than 2%, and if you're most probably will be getting favored regions in 95 and above. Okay, so your best thing for selecting your model will be Ramachandran plot. Okay, that is you have to come to here. After this, you have to come here. This one is based on the selection criteria. We have selected template in the first step. I think you remember that. You have selected template in the first region. So there are protein wherein you won't be getting percentage sequence identity. There, see here, I have taken, just going on to the first slide for the remembrance. Okay, here we have got e value 0. Okay, this is okay. This is a very common protein. Lactose D richness is a very common protein. So obviously we'll get homologous structures in the blast runs. Okay, there are proteins. You know, if you're running COVID or something like that, you won't be getting e values so closer to 0. You will be getting closer to 1 or something like that. In those situations, what has to be done is you can't be going for homologous modeling. You have to go for threading based methods. Okay, usually whenever you are selecting a template for your homologous things, it should be within the safe zone, and that is at least more than 50%. Okay, 50% homology has to be done. If it is in the twilight zone also, it is between 20 to 40. Okay, if it is in the twilight zone also, you will not be getting the perfect model out of it. And below that, it's midnight zone. You won't even get, whenever you won't even get homologous model out of it. The best solution would be, I told you the earlier slides, that if you're going for predictions, you can go for either homologous prediction if your structures are more than 50% homologous. Okay, and if your structures are falling within the twilight zones or mid zones, then you have to go for Rosetta. It's an online server. This Rosetta works on de novo threading procedures. It is a very complicated procedure. We can't be running in local machines and all. A server is there. Just put your sequence over there, wait for 24 hours or 48 hours. It will give you your best structure based on de novo sequencing. Okay, so this one, but most probably the structures which are predicted with Rosetta using threading methods will not satisfy Ramchandran plot everything, but we don't have any other option to predict a structure. If even structure fails in the Ramchandran plot, then no other option. You have to go for X-ray crystallography or NMR based structure prediction, which takes obviously more than 4 years. Okay, so this homology model only works for structures whose template match is more than 50. Your better thing is go for more than 70%. If it is going beyond that, just forget about it. You have to go for experimental predictions. Why do we go for homologous prediction is we will be able to predict the structure very easily like this, and we'll be able to do docking studies. We don't need to wait for extracrystalloid structures. We can go for docking studies and everything. Okay, I'll show you 2 case studies wherein we worked out very recently. We worked out. So this paper is by screening for curcumin analogs where targeting sortase A enzyme for Enterococcus faecalis. Okay, this particular Enterococcus faecalis is a very infectious organism wherein prediction going for structure prediction using X-ray crystallography or extracting the protein is a very tedious task. Okay, this sortase A is a very tedious task. So what we did was we went for homologous modeling using this particular thing. See here, we went for homologous modeling. We did, there are some procedure, whatever the scores whatever the procedures I told was for the basic protein. That if you are not getting here, what we did was we were targeting only the functional domain of that particular protein, and we are little worried about the other structures. Even if it is having more amount of disallowed regions also, we are little worried. So what we did was we did homologous models for sortase A, and we tried curcumin analogs as a ligand for this. Okay, so and we did a molecular dynamics studies for this particular thing, and we found out curcumin analogs were against this particular sortase A very effectively. And in the other paper wherein we had, okay, this is a paper called equivalent alkaloid potential modulates amyloidic structural transition of biofilm scaffolding small basic protein. This is from a Staphylococcus aureus also. This is also another infectious disease. So we won't be able to handle up all this in the lab that we are working up here. So what we did was we took up the mass spectroscopy data, and directly from that we started doing homologous models. Okay, in this particular, as I told, we did not find any particular homologous structure template. So what we did was we went for Roberta. Yes, we went for threading over here. It took almost several iterations to do this particular thing. So we went for Roberta, and then finally we energy minimized our structure. There are steps to do after this. So we energy minimized our structure for further docking, and then we docked our structures against camptothecin over here. So we found out this camptothecin was very effective against these particular things. If you think of this doing in your structural biology lab as experimental procedures, it will take years and years. And this work was entirely done using during the COVID period. Everyone was working online, so you make use of this 2 years for working on these papers, and we found this particular protein. This is the protein that we modeled out of it, and we used this for molecular dynamic studies also. And we found out camptothecin was very effective. Right now, we have extracted this protein in the lab in its campus, and we are working on this directly over there. Okay. So we need a lot of restrictions while working in infectious disease. That is what the lab that we work was an infectious disease. We can't be using these strains everywhere and all. So we had restrictions to use it. So whenever we go for any protein synthesis or something like that, we'll first initially go for in silico methods, and then after that we will be going on to the main things. Okay, so this is the end of this. I'm just stopping my slides. Any doubts you can ask me up. I'll be glad to speak up.
Divakar?
Thank you, sir, for your nice presentation.
Yeah, thank you, Divakar.
It is a complete presentation where we can learn both the introduction of the topic and the demo. Dear participants, many of you might not be able to follow what is the procedures I have used, but all these things will be available in the YouTube. Okay, you can, after the webinar, you can check it, and then you can download the software which was recommended by sir, and then you can practice. Okay, we will also share the email ID of our speaker, and if you have any doubts, he will be very well interested in helping you. Thank you,
yeah,
sir.
Participants, you can mail me anytime. I just put up in the chat also my email ID. You can mail me anytime. I'll be glad to do any doubt clearance sessions and everything. You just mail me up. I'll tell you the full protocols, because telling in a hour, it is not a problem. Okay, this procedure almost takes almost 3 hours to complete for a full day or something like that. Do a hands-on training of your own. If you have any doubts at any point of time, do contact me. This is my email ID, ram.k.pt@kc.ac.in. So you can email me up anytime.
Participants, you have any questions? Actually, one question is there, sir.
Yes.
Sir, from homology modeling, how we can select a binding pocket for docking from a homology modeling?
Homology modeling. See, when we are going for homology modeling, the best thing is this one is a very easier thing. You must have a template, or you must have selected a template for homology modeling. You can use the same template binding pocket as a binding pocket for a homology model protein also. Or if you are reluctant to use it, you can go for the server called CAST-P. Okay, I'm just putting up in the site there. It's a site called CAST-P, wherein it will predict the binding pockets and give it for you. Okay, so you can use that particular site for predicting your binding pockets, and you can use it.
Dear participants, are you having any questions? Okay, sir, then no questions. Thank you very much, sir.
Okay, Devagan, it's nice meeting you all. Thank you.
Dr. Ram Kodandan, sir, thank you so much. On behalf of the department, I would like to thank you. Your presentations was very informative, sir, actually, especially regarding the homology modeling. I think we should call you for a, in-person, meeting with all our scholars.
Yeah, I'm in Coimbatore only. You can call me anytime. I'll be glad to come to
we'll call you when we do a, offline conference.
Yeah.
So thank you so much for your acceptance and being here with us, sir. Have a good day, sir.
Yeah, sure, sir. Thank you, sir. Thank you very much. Okay, Devagan, I'll take a leave. Okay.
Thank you, sir. Thank you very much, sir.
Okay.
Okay, now we are coming to the end of the session. Our final speaker is with us, Dr. Praveen Takikuppe Krishnamurthy, head of our department, JSS College of Pharmacy, Pharmacology Department. He has completed his bachelor's degree and master's degree from Rajiv Gandhi University, Bangalore. He obtained his doctoral degree in pharmaceutical sciences from the Tamil Nadu Dr. MGR Medical University, Chennai in the year of 2019. Currently, he is working as a professor and head for the Department of Pharmacology, JSS College of Pharmacy, Ooty. He has expertise in nanomedicine, molecular biology techniques, pharmacological techniques, computer-aided drug design, chemical synthesis, and isolation of phytoconstituents. His core research areas include metabolic disorders, cancer research, and neuropharmacology. In all those areas, he has been developing targeted nano drug delivery systems to improve drug molecules pharmacokinetics, efficacy, and safety profile. As a PI, he has received a total of 286 lakhs in research grants, including 3 national research grants from the Department of Science and Technology and Department of Biotechnology. He also received 1 international grant from the Department of Science and Technology in Indo-Uzbek Joint Research Project. With this information, I invite him to share his presentation. Please welcome, sir.
Thank you, Devakar, for the nice introduction. I take this opportunity to welcome all the participants for this presentation on animal testing in biomedical research, history and alternatives. So before we go into the history of animal testing and the various alternative methods, we'll first understand why animal experimentation in biomedical research. These are some of the reasons why we use animals in biomedical research. The first is to satisfy scientific inquiries. If you have an hypothesis, you want to prove or disprove, you try to use animal models. Animals are used as a model to study human anatomy and physiology, and animals used as a model to study disease. We can induce a number of diseases which occur in humans, and also we can mimic in animals. Therefore, we basically use them as a model before testing any drug in humans. Animals are also used to develop and test potential forms of treatment. And another very important applications of animals in biomedical research is in the safety studies, where we use animals to protect the safety of people as well as animals as well as the environment by testing the toxic profile of xenobiotics. Coming to the recorded history of animal testing, I would say the word recorded history, not the real history. Nobody knows why animals have been used, when animal usage was started. Therefore, whatever the recorded history available, it dates back to 600 BC to the Greek. So where Greek philosophers like Alcmaeon was the first person to have been used animals for understanding human anatomy and physiology. During those days, people started using animals either because of curiosity or maybe because of religious restrictions which prevented them dissecting human body. People thought that if you dissect human body, they have next incarnation or next life cycle where their body may get mutilated. Therefore the human body dissection was not allowed. Therefore, as an alternative source, people started using animals. Inspired by the work of Alcmaeon, another Greek philosopher, we all know Aristotle, in the 400 BC, extensively worked with animals to understand anatomy and physiology. So his work resulted in 2 important publications called Histories of Animals and Parts of Animals. Then in 300 BC, Galen, a Roman philosopher, first time shown the in a scientific way demonstrated the dissection technique or the vivisection techniques in animals. And later on in the 16th century, Francis Bacon, who is considered as a modern father of modern scientific methodologies, in fact, done a lot of work to understand the human anatomy and physiology, where the work of Francis Bacon, in fact, made, in the 16th century to people appreciating the vivisection as a method to understand human anatomy and physiology. Coming to the 17th century was considered as a age of enlightenment and scientific progress, where the work of English philosopher William Harvey was very prominent. William Harvey worked with both humans as well as with animals to understand the circulatory system. So before his work, it was believed that liver produces blood continuously for the consumption of all the organs, and heart was considered just as a heating chamber, and arteries carry air and veins carry blood. So William Harvey's work with animals, in fact, changed the entire understanding of circulatory system. In fact, he gave the most accurate description of the circulatory system what we study today. So therefore, 17th century is considered as an age of enlightenment. Then came the work of Stephen Hales and Van Haler, where they have worked extensively in the 18th century on inflammation process, neurophysiology, and heart function, as well as on hemodynamics. The Van Haler was the first scientist to measure blood pressure, and also he is the one who discovered forceps. At the end of 18th century, there was a raise of moral consideration for animals. Before this, there was not much, importance was given to the animal moral consideration or animal well-being in research. So people before believed that, like earth and water, the God has created animals also for the consumption of humans. So in this context, there was not much, consideration was given, moral consideration was given before. But later on, in the 18th century, things have started changing, where people started believing animals are also like humans, do feel pain and distress. Therefore, they should be treated humanly, and they should not be subjected to undue cruelty. So in the 19th century, uh, there was a upsurge of animal experimentation. We will discuss about why there was a significant rise in the animal experimentation. And later on, there was an important discoveries happened in the 19th century, like pasteurization technique was discovered, rabies vaccine was discovered, anthrax vaccine was discovered, and polio vaccine was discovered. Some of the important discoveries were happened in this duration. And the rise of animal experimentation was in fact strongly promoted by French philosopher, physiologist, in fact, Claude Bernard. So Claude Bernard was considered as the initiator of experimental animal research. So Claude Bernard for the first time described the difference between observers and experimenters. So according to him, observers are those who just observe the nature without fiddling with it, whereas experimentals are those who deliberately alter nature to seek natural phenomena. In his terms, he wanted to promote people to do experiments by using animals. The whole idea of him promoting experimentation with animals is to criticize those days physicians who used to do uh, surgeries in humans, and people used to die due to surgical, post-surgical complications like infections. He used to demonstrate the same thing in the animals to criticize the physicians. His idea was promoting usage of experimental animals in research was to criticize physicians during those days. Later on, the work of Louis Pasteur and Robert Koch resulted in a lot of discoveries like pasteurization and rabies vaccine. Louis Pasteur worked extensively with rabbits and dogs to discover rabies vaccine, and so on. Many important discoveries have happened. So later on, in the year 1875 in UK, the first anti-vivisection society has what happened? Lights are not moving. Okay, but thank you. Now you check. Is the slides are moving now, Divakar? Now it's moving.
Yes, sir, it's moving, sir.
Previously it was moving.
Previously, it was not moving, sir.
Okay.
Admit it. Dear participants, please wait. That there is a technical issue. It will be resolved within 1 minute, 1 or 2 minutes.
Can you hear us now? Okay, but thank you. Yeah. 20th century is considered as triumph of uh, science-based research, where important discoveries like chemotherapy, vaccines, blood transfusion were all discovered, and also thanks to Russell and Bush, where they made alternative concept as a popular concept, where people started using alternative methods for the instead of using the animals in research. And thanks to Bruce and Ames, who discovered Ames bacterial reverse mutation assay as an alternative method to usage of mouse in mutation studies. And if you see the timeline of medical research, you will appreciate that important discoveries like blood transfusion, MRI technology, heart transplantation, and various vaccines were all discovered through usage of animal experimentation. And therefore, you will appreciate animal contribution in more than 70% of the Nobel Prizes, which have been received in the area of physiology or in medicine. So if you see the timeline of usage of animals in medical research, biomedical research, you will appreciate there is a significant rise in the animal usage in the 19th century or in the 20th century. So this is because of the two important drug tragedies which were very unfortunate. The first one is the 1937 elixir sulfonamide tragedy, and the second one was the 1960s thalidomide drug disaster. So these two tragedies made uh, uh, drug testing in animals a mandatory process in the drug discovery and development process. So the first tragedy, the test of raspberry, taste of death, the 1937 elixir sulfonamide tragedy. So in 1937 in USA, this tragedy has occurred, and during 1937, so in USA 1906 FDA Act was enacted, and this act was old act, and it was considered as useless because this act was only monitoring the label claims. During those days, there was no formal approval was required for marketing a new drug for pharmaceutical industries. So during this period, German scientist Gerald Domack discovered sulfonamides. These drugs, unlike now, were considered as a wonder drugs because of their very good effectiveness in treating pneumonia, blood poisoning, and meningitis. But unfortunately, these drugs, the sulfonamides, were available only in the powder formulation. And there was you all know that most of the drugs are bitter in nature, as in case sulfonamides also. As a result, doctors started demanding the pharmaceutical industries to come out with a syrup formulation or the elixir formulation of the sulfonamide drug. So one of the companies which was marketing sulfonamide powder formulation was SC Messengers from Tennessee, USA. This company asked the chief pharmacist to come out with the elixir formulation. And the chief pharmacist, tried to dissolve sulfonamides in various solvents, but unfortunately, he found one solvent which is able to dissolve sulfonamides was diethylene glycol. Little he knew that diethylene glycol can cause tragic, uh, drug in toxic incidences. But however, he did not know, as the 1906 FDA Act did not mandate the safety studies. So for the chief pharmacist quickly made a formulation, and he added a raspberry syrup, and he made an elixir formulation, and this was dispatched to the all across the US. And after consumption of this drug formulation, more than 100 people died because of the kidney failure. And so the US government has acted quickly and asked two scientists, Gehling and Kennan, to investigate the cause for the death. And they, in their report, found that the solvent which was used, ethylene glycol, was responsible for the kidney failure. And later on, in addition to this, they also proposed principles of drug testing before trials in humans. So according to their proposal, the following information need to be generated before testing any drug in the or a new formulation in the humans. And so, like, exact composition of the drug should be known, acute toxicity data, chronic toxicity data, drug interaction data, and so many other information need to be generated. So in animals before testing in humans. So the same principles are also holds good today. We also do the same thing now also. So immediately after this tragedy, US government, introduced the pending amendment to the 1906 FDA Act, that is the Federal Food, Drug and Cosmetic Act 1938 was enacted. This new law gave the FDA more powers to monitor the safety as well as the efficacy of the drug. So with this tragedy, the usage of animal experimentation was strongly increased in the USA. But however, the same was not enacted in the other countries like European countries. So unfortunately, exactly 25 years later, the another drug tragedy, thalidomide drug disaster, was repeated in Europe, especially in Germany and England, where this was considered as one of the greatest drug tragedies. So thalidomide was a tranquilizer, and which was used for as a painkiller and was considered as wonder drug those days. And unlike now, those days off-label prescribing was very common. For example, if a doctor feels paracetamol may be useful for some other indication, maybe for treating, take a crude example like diarrhea, is free to prescribe. So one of the Australian physicians, found this thalidomide useful in the treatment of morning sickness in pregnant women. So this became very popular in Europe, and European physicians started prescribing thalidomide for the management of morning sickness in pregnant women. Little they knew that thalidomide causes teratogenicity, and as a result of this, more than 10,000 children all across Europe, out of around 46 countries, were born with organ malformalities, and this has further made drug regulatory agencies to make toxicity testing as an important requirement for drug discovery and development. And as a result of this tragedy, one more amendment to the FDA Act was enacted, that is called Drug Efficacy Amendment Act of 1962, which is also called as Kefauver-Harris Amendment to Food and Drug and Cosmetic Act. And this act made it mandatory for the drug manufacturers to provide efficacy data in addition to safety data, and also manufacture drugs according to the good manufacturing practice, or the GMP level. So with the rise of animal usage in drug discovery and development, there is also an opposition to the usage of animals. So people who oppose the usage of animals had two views. Some had abolitionist view and some had welfarist view. Those who had the abolitionist view, they wanted complete ban of animals in the experimental research. They believed that animals and humans are entirely different, and the data generated in animals is misleading and may not be of any use. But however, those who oppose, who had the welfarist view, they believe that usage of animals are important in enhancing the knowledge of human anatomy and physiology, and also discovering novel drugs for the treatment of human diseases. But however, they wanted animals to be treated very humanly or fairly during the experimentation, in the sense, so animals has to be given utmost care and respect while doing the experiment, with minimization of pain and distress. So those who oppose animal testing, because of the want of time, I may not be reading, believe that benefit to humans does not justify harm to animals. And those who favor animal testing believe that experiments on animals are necessary to advance medical and biomedical research. And both of their views can be summarized with these two pictures. So in the left side, we can see the cruelty on the animals, whereas in the right side, we can see humans and animals would still be dying from rabies if Pasteur hadn't experimented with dogs. So both the views are right. So now what is the middle path would be is to balance both, where we use the animal for experimental research. At the same time, you try to treat the animals in a human way possible by minimizing pain and distress during the experimentation. Thanks to Russell and Burch, so who made alternative methods as one of the popular methods. So in 1950s, University Federation of Animal Welfare has asked Russell and Burch to discover or to find out the ethical aspects of animal research. So in their research, they came out with their findings in the form of a book called The Principles of Human Experimental Techniques by Russell and Birch in the year 1959. In that book, for the first time, they proposed the 3R concept, that is the replacement, reduction, and refinement concept. So this concept has provided the blueprint for combining animal welfare considerations and quality of research. And also this concept has become the central theme for growing anti-vivisection societies, where even today in the institutional animal ethics committees, we apply these principles of 3 Rs, that is replacement, reduction, and refinement. So what are these 3 Rs? I think most of you may be knowing. Replacement, wherever possible, replace animal experimentation with non-animal methods or with less developed animal species. So what is reduction? Reduction means reduce the usage, the number of animals as much as possible without compromising the quality of research. In simple words, instead of using 10 animals per group, if you can get the same quality data with 5 animals per group, you try to reduce the usage of animals to 5 animals instead of 10 animals per group. Refinement refers to use all those possible interventions to minimize pain and distress to the animals so that animals can be treated humanly. So what is the rationale for using these alternative methods? So the reason why we need to use alternative methods is based on some of the facts. It's not just because of the ethical point of view, it's also based on the scientific reasons. Many times it's not that animal experiments provide the utmost quality data. It is many times it is the in vitro methods or the alternative methods which generate better quality data than the animals. So some of the reasons which we can list is, according to a conservative estimate, every year around 12 million animals are used in experimental research. So according to FDA data itself, 9 out of 10 drugs deemed to be successful in animals test fail in human clinical trials. So therefore, animal validity of animal data is only assumed rather than proven. So, and also animal testing, is demanding with regard to cost as well as time. So therefore there is a growing acceptance that non-animal research methods are advanced techniques with fewer demand on the time and finance. So why non-animal research methods sometimes are advanced technique? For example, if you take cell culture methods, will permit us to see what is happening at the cellular level or at the molecular level, which was not possible at the whole animal level. Therefore, non-animal techniques many times are advanced than the animal techniques. Therefore, we should always try to use non-animal methods as well as much as possible. So coming to the alternative testing methods, some of the alternative methods which I will be discussing briefly include epidemiological studies, in vitro human cell and tissue cultures, clinical studies, computerized patient database and post-marketing surveillance, mathematical models, computational simulations, autopsies and post-mortem studies, non-invasive imaging techniques, use of invertebrates and lower vertebrates as an alternatives, and also some alternatives in education. So let us quickly go through epidemiological studies. Epidemiology is the study of how often a disease occurs in a group of individuals and why. So epidemiology tries to establish a correlation between the exposure of a substance to the adverse health effects. So epidemiological information will help us to plan and evaluate strategies to prevent illness and manage disease. So epidemiological studies are non-invasive studies. So we can always group people based on their exposure. For example, if you want to analyze the exposure of smoking, whether it leads to lung cancer or not, we can always follow group people into smokers and non-smokers and do a long-term follow-up to identify how many people who are smoking will develop lung cancer and how many people who are non-smokers will develop lung cancer. By analyzing this data, it is possible for us to come to meaningful conclusions without even need of use of animals. So like this type of epidemiological studies are very meaningful and very effective because the data is generated in humans. There is no need for usage of animals for generating such meaningful data. So this is how we can save usage of animals in experimental research. So another very important alternative is the in vitro human cell and tissue cultures. So in vitro human and tissue cultures will help us to understand pharmacodynamics, pharmacokinetics, as well as toxicological aspects of the drugs. So cell lines and tissue cultures will help us to do this. And many times they do have a very good correlation in generating the data with what we could generate in the whole animal. In this diagram, we can see the in vivo clearance correlation as a strong correlation with regard to the whole animal study as well as with regard to the cell lines. Some of the in vitro alternatives which have in fact replaced the animal studies, especially in the eye irritation corrosion studies and skin irritation corrosion studies, where previously we used to use rabbits for this. Nowadays, we are not being permitted for usage of, these animal studies for skin irritation, corrosion, or eye irritation, especially for cosmetics. So we all should have a alternative method. So many alternative methods have been developed in this regard. Some of the in vitro eye irritation alternative methods include red blood cell test, and then episkin and epiderm are some of the tests which are developed for in vitro skin irritation as an alternatives. And 3T3 NRE phototoxicity assay is one of the assays which was developed as an alternative to phototoxic testing. So epiderm or episkin is an alternative where we can grow keratinocytes in the in vitro and test the drugs in the in vitro cell culture to find out their toxicity, to identify whether they are having a corrosive or the irritation potential. Similarly, 3T3 NRU phototoxicity assay can help us to identify phototoxic potentials by in a 96 well plate instead of doing the whole animal toxicity testing. Another very important in vitro alternative is on organ on a chip. Organ on a chip is a microscale system that is used for mimicking the human body environment. Different organ on chip, liver on chip, or kidney on chip, or heart on chip can be developed where they are based on the principle of microfluidics. So normal cell cultures are two-dimensional in nature, whereas here the cells grow in a three-dimensional way by using the microfluidics. The use of microfluidics mimics the flow of blood, and connecting different organ on chips, we can develop an entire organism, or we can create a whole human body system where we can test the drugs or inject the drugs into this microfluidic system to analyze the pharmacokinetic aspects, pharmacodynamic aspects, or the toxicological aspects of the drugs, mimicking the whole animal system. A lot of work on, a lot of importance has been given to organ on chips so as to develop or mimic the human anatomy and physiology in the in vitro environment. So next type of alternatives is the clinical studies. So careful design of clinical studies, like previously we studied one clinical study, that is the epidemiological studies, can test the drugs in the in vitro cell culture without use of find out their toxicity studies to identify. So another very important clinical study is the studies potential. Where similarly 3T3 we can test directly the drugs at a very small concentrations, or we can say subclinical doses. That is why the word micro. Micro refers to very minute concentration of the drug. So this drug is given to the humans directly, and where the drug does not produce any toxic effects or it does not produce any clinically visible changes. So such doses, however, can produce biochemical changes. By analyzing these biochemical changes, it is possible for us to come to meaningful interpretations. So this type of studies are called microdosing studies. The next type of studies are the autopsies and postmortem studies as an alternative to animal testing. So autopsy studies are responsible for discovery of and description of many diseases. So autopsy tells us what is the cause of the death, what was the state of the individual before he died, and whether any medical diagnosis or the treatment given was appropriate or not. So one of the important application of autopsy studies is understanding the neurological disorders, brain banks of a dead patients suffering with some particular disease like Alzheimer's disease or Parkinson's disease. The study of such brain banks has helped us to understand the pathology behind the Alzheimer's disease and come out with newer intervention strategies to treat these diseases effectively. So computerized patient databases and post-marketing surveillances are another very important alternative methods which can replace animals. So data is money, and computers can help us to organize data in a systematic way. And analyzing this data, large amount of data in a systematic way, can help us to identify the side effects of the drug, and newer indications of the drug can be identified. For example, post-marketing surveillance of side effect analysis of many drug data has resulted in identification of new indications for many drugs. For example, anti-cancer properties of nitrogen mustard and actinomycin D were all identified like this, and also mood-elevating properties of tricyclic antidepressants were also identified in this fashion by analyzing the data obtained post-marketing. So mathematical models and computer simulations in silico methods are one of the very important alternatives and very popular alternatives as well. I think you would have learned about the homology modeling in the previous this one, and also previously you talked about the previous speaker also spoke about the molecular docking or the various computational techniques. So computational techniques will help us to screen large database of molecules without even doing a test. QSR methodologies will help us to identify the potency of a drug molecule without even testing them in a in vitro or even in the in vivo system. And computational methods will also help us to predict the structure of the proteins, like homology modeling can help us. So as a result, so computational methodologies have huge applications in drug discovery and development as an alternative to animal testing. So non-invasive imaging techniques like CT scan, MRI can also be used as a alternative methods, because these technologies have got a ability to resolve the structure into a single cellular level. Therefore, they permit us to study directly in human organs what are the structural defects which are happening or the functional defects which are happening. Therefore, help us to design better interventions. So use of lower vertebrates or invertebrates as an alternatives. So this may raise a question here for you, why I'm suggesting a smaller animal species as an alternative to a larger species. So this is based on the concept that animals which are having underdeveloped nervous system perceive pain less than compared to an animal which is having a well-developed nervous system. For example, if you compare zebrafish and a dog, you will not see emotions being expressed by a zebrafish, whereas a dog can express emotion and pain in much more way than a zebrafish. So in that context, so replacement of a larger animal with a invertebrate or a lower vertebrate is always considered as an alternative. So therefore, a primate may be replaced with a transgenic mice in polio vaccine development, or Drosophila or sea lichens or zebrafish can all be considered as an alternatives to mice or rat, or even to the primates or to the dogs. Coming to the alternatives in education, we use many alternatives in education, and it is believed that many times the students learn better by using alternatives than doing a live animal dissection, because alternatives permit the students to repeatedly practice things or also clear things, clearly visualize things better when compared to doing a dissection in live animal. So some of the alternatives you already know, virtual, experiments where you have XFarm, Xcology, and all these softwares are available where we can do many experiments what we routinely do in the experimental research in pharmacology. And with regard to the regulatory agencies, there are many regulatory agencies have accepted the alternative methods. So acute toxicity studies have been now replaced by alternative toxicity testing methods like acute toxic class method, fixed dose procedure, up and down procedure by the OECD. And these are all considered as alternatives because we apply the principle of reduction here. So when compared to conventional alternative methods, for eye corrosion testing, we have got bovine corneal opacity permeability assay instead of rapid testing. And isolated rapid eye test is also one of the methods, ex vivo methods. For genotoxicity, Ames test is one of the most popular, and there are so many other tests which are available. Like that, we have got phototoxicity testing has been replaced by alternative methods. Pyrogenicity testing has been replaced by alternative methods. Skin corrosion testing has been replaced by alternative testing methods. So these are some of the alternatives which I prefer to discuss with you all today. So coming to the challenges faced by the alternative methods, of course, a lack of focused research is one challenge, and also availability of funding, and also availability of computational databases for scientists to access as an alternative source material for doing research, and lack of development of technology. For example, we do homology modeling, prediction, and all these things by using computational techniques. However, till date the technology is not good enough to predict the biology in a more accurate way so that we can develop the drugs. For example, if you develop a QSR equation, if it becomes very accurate, which can mimic the what happens in the in vivo, then we don't have to do any testing. All QSAR equation will tell you the potency of a drug. We can directly use, we have QSAR equations for predicting the toxicity, everything. But unfortunately, biology is so complex that current technology is not able to predict or develop an equation which can predict the biology in an accurate manner. So these are some of the challenges, which are facing today for developing the alternative research. So in conclusion, I would say that it is strongly believed that the experimental animals may be eventually replaced by more advanced alternative methods in futures. However, current alternative methods are still technologically immature to replace everything at this moment. So I would like to end my talk by the quote of Mahatma Gandhi ji and Nobel laureate Albert C. Schweitzer. Thank you so much for your patience listening. If you have any questions, I would like to take it up.
Thank you, sir. That was a very good information you have shared with us regarding drug toxicity and alternative to animals. Yes, sir, we agree with you. We need to reduce the animal cruelty, sir. Audience, please, if you have any questions, unmute your mic and then you ask to the speaker. Participants, any questions?
Good afternoon, sir.
Good afternoon.
Sir, can you give some more information regarding the organ on chip method?
Yeah. See, organ on chip is a microfluidic based methodology where, see, what happens in a cell culture, if you see 96 well plate, if you plate the cells, what happens? The cells grow in a two-dimensional. So on the bottom, it will not grow in a three-dimensional fashion. But however, the cells in the human body, they are all in a three-dimensional space. Okay, they will be interacting with the neighboring cells in a three-dimensional pattern. So this can be mimicked by using organ on a chip, where the microfluidics, it is based on the microfluidics, where the fluid will flow through the cells, and it is based on a chip-based system, like your mobile SIM chip. So here we can grow the cells, and these here the cells will grow in a three-dimensional fashion, and they mimic more of a human condition than a in vitro cell culture condition. So you can develop different organs on a chip. For example, heart on a chip, liver on a chip, kidney on a chip. So these chips can be connected to each other to mimic entire human body. Okay, you can incorporate all chips containing the human organs, and you connect them, and then you can test the drugs in them for identifying their pharmacokinetic profile, pharmacodynamic profile, and toxicological profile. This is how organ on a chip can help us to understand how the whole human body reacts to a drug without even requiring to use an animal.
Pharmacokinetics also can be studied.
Pharmacokinetics can also be studied, yes.
Yeah. Thank you, sir.
Right.
Any more questions, dear participant? Okay, sir. Thank you, sir. Okay, sir. We come to the final session. We end of the session. Okay. Now I will request Dr. R. Vadivelan, sir, Professor, Department of Pharmacology, JSS College of Pharmacy, to deliver vote of thanks.
Thank you, Dr. Devadar. Good afternoon, ladies and gentlemen. We have come to the end of session. It's my responsibility to deliver the vote of thanks. First of all, I thank today's resource persons who gave the wonderful lectures on the popular lectures series on biotechnology with the titled Applications of Proteomics in Biomedical Drug Discovery. First, I'd like to thank Dr. Jaikant Chandrasekharan, the Associate Professor from Sri Ramachandra Medical College of Porur, who delivered his talk on multimodes and dimensionality reduction in drug discovery. Thank you, sir. Secondly, I would like to thank Dr. Ram Kodandan, Assistant Professor from Kumaraguru College of Technology, for his wonderful lecture on from sequence to structure and in silico drug discovery. On behalf of Department of Pharmacology and JSS College of Pharmacy, wholeheartedly thank you, sir. Next, I would like to thank Dr. T.K. Praveen, Professor and Head, Department of Pharmacology, JSS College of Pharmacy, Ooty, for his wonderful lecture on animal testing in biomedical research, history and alternatives. So wholeheartedly, I thank you, sir. Next, I would like to thank Dr. S.P. Dhanapal, the principal, for his encouragement and support throughout this popular lecture series on the biotechnology. I'd like to thank my department HOD, Dr. Praveen, and all the colleagues for their moral support in this DBT-sponsored webinar series. And I would like to thank the program coordinator, Dr. Divyagar, and his team, along with the PhD scholars, for their wonderful completion of this DBT-sponsored webinar. And I would like to thank the technical support rendered by Mr. Jayakumar C for conducting the webinar in a well smooth manner. I thank you one and all. Thank you.
Thank you all participants. This is the end. This is the end. You can leave. You will receive all your certificates through your email ID, which you have used for registration. Thank you. Thank you all.
Thank you.
Okay. Okay.
Thank you, sir.